that the benefit still outweighs that risk in terms of mortality. But again, it comes back to the idea of the most potent drugs we have are food and exercise.
Absolutely. And it comes back to me as well that it's not about how many years we live, our longevity, and it's how are the last years that we live, right? And if statins are going to come back to haunt you in 20 years because they're going to have extra or increase in diabetes, for example, yeah, it might buy you extra time now. But again, if you have food and exercise as your main medicine.
The Zone 2 training for me, it's just become such an important part of my training for myself and for my patients. A question I get asked a lot that I don't know the answer to, so I'm going to ask you is, what's the minimum effective dose? Because obviously, I would love it if I could wave a magic wand and have 1 hour per day to do Zone 2, and then on top of that, layer in all other exercise. That would be amazing, but it's not. I only do 3 hours a week of Zone 2, typically in 4 45-minute to an hour sessions. Do you think that's enough?
Yes. So this is what I've seen and I've learned from the athletes. And I would love to do this now with patients. What's the right dose? But we know, or at least I've seen with athletes, that if you do that 2 days a week, one is the dose and the other thing is the frequency. So if you do that 2 days a week, you maintain. And we see athletes who, in the off season, cyclists, for example, or runners or triathletes or swimmers or rowers, if you do the Zone 2 5 days a week, for example, you really push the needle. Then once the season starts, you need to do more higher intensity exercise and training. And then you have the races and you need to recover. So definitely you cannot do Zone 2 every day. So what we see is like 2 days a week, it tends to maintain.
So that's the frequency. What's the dose?
Yeah, and the dose, what I see is like, obviously these elite athletes, they need to keep pushing the needle. 1 hour is not going to do much for them. Yeah. Because they have that stimulus already, or 2 hours. They might need 4 or 5 hours. But a patient with type 1 diabetes, maybe 1 hour is enough. And that's what I'm trying to fine tune, what would be. What I know very well is that 3 days a week, it starts moving the needle, 4 or 5 for sure. And what I've seen or guessing as because we don't have any real data, this is about 1 hour to 1 hour and a half. It does the trick for those who have type 2 diabetes or pre-type 2 diabetes, for example. So we have last year a patient who was diagnosed with that's what we were saying is like late pre-type 2. What the heck is that, And then with 1 year, doing an hour into an hour and a half, 4 days a week, she reversed that completely.
Okay, that's a pretty big dose. So, I mean, for me, it's always for me Tuesday, Thursday, Saturday, Sunday is Zone 2. It might be that those Saturday, Sunday workouts, I need to push them longer. Maybe I need to do 90 minutes on each of those days and stay at 45 minutes on Tuesday, Thursday.
It could be, but at the same time, it might be your right dose because you're not in that unhealthy population side. So your dose might be lower.
But my thinking now is that this is such an important part of cellular longevity that this is the difference between being a healthy 90-year-old and being, in my framework, it's 1/4 of the equation. What would you do?
I would do, and this is my case, when I stopped cycling, what I told you earlier, right? I gained 65 pounds because I was working 70 hours a week and exercising 6-7 days a year.
And still eating like a cyclist?
And eating like a cyclist. And I'm from the Basque Country, and we like to eat food because it's one of the best areas in the world. And probably also I had insulin sensitivity developed from I was a cyclist, which I would just pour carbohydrates, and then I would not burn them. So maybe I just transform more into fat. I also have a familiar dyslipidemia. So I have high triglycerides and high cholesterol genetically. So I didn't take care of myself. I would not exercise and eat a lot. So I gained 65 pounds in about 8 years or so. And then I said, wow, I went and did myself a checkup. And then my blood pressure was 125. I was in my mid-30s, 125 over 85. So was getting there. And my triglycerides, once I saw them 800, which is huge, off the chart, right? Back in the days, people didn't do A1C. That's when I started to work on these concepts too. And so I started to apply this to myself. So I started doing 4 days a week. Even 1 hour was, I was bunking because I was not used to that. It was very depressing going to
I was about to say, that must have just been devastating.
Oh my gosh.
To go from being a professional cyclist to struggling to do 4 hours a week of cycling.
Yeah. And knowing the same roads that you go to and that you couldn't go up the hill. But I lost 35 pounds within 7 months.
Did you make much change to your nutrition?
Exactly. I I was willing to eat a little bit less, but not sacrifice many things. Because again, for me, nutrition is very important from my culture standpoint. I love chocolate. I love wine. I love pasta and bread. It's ingrained in my culture. I was not going to renounce to these things. So that's what I said, okay, I'm going to try to give it a shot.
So you weren't going to go on a ketogenic diet.
No.
And it's an interesting example of there's give and take. Yeah. The more you're willing to push this type of training, the more you can keep on the other side versus, I've seen more patients than I can count with type 2 diabetes not exercise at all, but go on ketogenic diets. Within 6 months, they're off insulin. Within a year, they have a normalized hemoglobin A1C. But again, so it's almost like two levers. How hard are you willing to pull on each of the levers?
Exactly. No, that's a great comment for sure. And I think it's the debate. For some people, giving up chocolate is not a problem. For me, it's death. I just love chocolate. It's not that I eat it every day, a whole bar, but it is, or bread, That's one of the things too that is the balance. I lost 33 pounds and I stopped there. I could not lose more than that. I needed to then increase my dose. So I went from 1 hour to an hour and a half, 4 days a week. I lost another 10 pounds. So I lost a total of around 50 pounds, 47 to 50 pounds. That was 11 years ago before coming here. And I kind of keep it like that.
Now, this is interesting. As you're probably aware, the exercise and weight loss literature suggests that exercise alone is not sufficient for weight loss. I've always wondered if that was an artifact of the fact that they're studying exercise incorrectly, that the prescription, it's either the dose or the frequency or the intensity were not optimized. You were doing a very specific type of exercise. You were not exercising for the number of calories you burned. You were training your mitochondria to become better at fuel partitioning. That's a very technical description of what you did. I think it's important for people who are listening to this to appreciate that nuance. You were not there calorie counting, saying, okay, I'm doing 6 hours a day at this many calories, because you can achieve that in many different ways. It was almost the maniacal specificity with which you approached this that you basically said, you didn't think of it as I'm exercising 6 hours a week. It's I'm doing mitochondrial conditioning or reprogramming 6 hours a week.
Exactly. Yeah, I believe so. And that's what we know now with patients when we study in the laboratory, that they always tell you, I always train at this intensity, and you know that at the intensity they burn 0 grams of fat. They burn a lot of calories, but 0 are derived from fat.
They're actually working too hard.
And eventually, number one, you don't burn much fat. You burn fat in the post-exercise because you might increase your metabolic rate. But can that override the fat burning from the exercise itself? And second, it's too hard. You haven't exercised in a long time to start with, and you get into these high-intensity programs that they might not suit you or they might injure you. And many people give up. We see the rate of people giving up from gyms is about 50% or so within X amount of months. They either give up or their adherence decreases a lot. So when I ask these people who get into this extreme either exercise or diets, I always ask them, and they're successful, I ask them, can you do this for the rest of your life? And the question is, hell no.
Yeah, if you can't do it for the rest of your life, you have to come back to the why am I doing this? Using an extreme example, to do hill repeats up Alpe d'Huez, can you do that for the rest of your life? No. Yeah. Can you do it if your goal is to win the Tour de France? Yes. You're going to do it for 5 years. You're going to train that hard for 5 years. You're probably going to take a chunk of time off your life, by the way, but that's your job. You have to be the best climber in the world.
Exactly. And to the point of the nutrition is a must. You need to do something with it or do a lot more exercise. But I think it's the balance that we all, I think, need to understand better.
Well, that's for me why fasting has become so important. Now, you were laughing at me earlier before we started recording about how crazy it is that I can do these long fasts. But in many ways, it's a way to provide me balance. It's like sprinting. It's basically every month, there's just a frequency with which every quarter I do one type of fast, and every month a different type, and every week a different kind. It allows me to keep a balance, and it allows me to say, yes, I could do this for the rest of my life, whereas the reality of it is I couldn't do a ketogenic diet for the rest of my life. As powerful as it was in me, couldn't do it the rest of my life. So a couple other questions I want to ask you about. You've alluded to cancer twice now. We've spent a lot of time talking about type 2 diabetes as a disease state in which the mitochondria are not functioning well, and they provide this great contrast. But you've touched briefly on cancer. Is there any evidence that a patient with cancer has a higher lactate level on account of the fact that they have mitochondria that aren't working as well, the same way that the person with type 2 diabetes has?
So it is a fact. In 1923, almost 100 years ago, Otto Warburg from Germany discovered the transformation of a normal cell into a cancer cell at the metabolic level. And the characteristic of cancer cells was that they use a lot of glucose for energy purposes. Now, they use a lot of glucose. Back in the days, there was no genetics or anything. But what struck Warburg is the amount of lactate that they produced.
Was that what struck him more than the fact that they use so much glucose even in the presence of sufficient cellular oxygen and insufficient ATP demand? So it was more the lactate accumulation?
It was more the lactate accumulation. And that's why he came to conclusion that cancer was a metabolic disease caused by an injury of the respiration system in the cell, which is the mitochondria. And that's what was the thought for many years because of the lactate. Even before glycolysis was invented, Meyerhoff, who discovered glycolysis, sometimes it's called Emden-Meyerhoff pathway, which is glycolysis, Meyerhoff was a student of Warburg. Before they had even found out about glycolysis, the way they measure glycolysis is by measuring lactate. So they would measure how much lactate the cell produces, and that's where they would say, wow, they're using a lot of glucose. But what he saw in cancer cells, there was an aberrant amount of lactate production. And that was one of the things that struck Warburg the most. And now what we see is that lactate is a typical feature of cancers. Cancers produce a large amount of lactate, which is also responsible of the famous microenvironment that a lot of people are talking about nowadays. The lactate microenvironment, the microenvironment of cancer cells is more acidic than non-cancer cells, and it's a niche for carcinogenesis. The responsible for that microenvironment is lactate. And what we know is that, yes, this lactate, it's a fact. There are multiple studies showing that, every study trying to find lactate in cancer, they're going to find higher lactate levels.
So this would suggest three distinct but not necessarily mutually exclusive explanations for the Warburg effect. The first being what Warburg proposed, which is in cancer, there is an injury to the mitochondria. As a result of that injury, the cancer it takes an inefficient path to go. Then there's the 2009 explanation proposed by Thompson, Cantley, van der Heyden in that science paper that was sort of a very important landmark paper that said, no, that's probably not it. It's the glycolysis and the lactate production is a byproduct of metabolic demand for building blocks. It's the cellular nucleotides that are necessary to build the cells. So the mitochondria work okay. What you're seeing is a deliberate and obligate choice to grow, and the need to grow, literally from a mass balance perspective, requires taking this pathway versus that pathway. And now you're saying, well, a possible third explanation is the cancer relies on lactate as a signaling molecule. And again, these could all be true on some level. We know that they're not all always true. I mean, at least we know that in the case of the Warburg effect, that's not universally true, that the cancer damages the mitochondria. Yeah. What are the next steps in figuring this out? And perhaps more importantly, much more importantly, how do we use this information therapeutically?
Yeah, so one of the things is that we have just finished a study. It's under review now, and we're going to replicate it now with more cancer cells. But we have done a study with the MCF7 cancer cells, which are the most common type of breast cancer, or one of the most common type of breast cancer cells. And what we have seen is we expose the cancer cells to glucose. So we did an experiment. One, exposing the cancer cells to a media that contains nothing. No glucose, no glutamine, which is also highly expressed in cancer. And these can survive a couple of days in that state. Then in another experiment, we just expose them to glucose. That's it. And in the other two other experiments, we added to that glucose media, we added 10 millimoles of lactate and 20 millimoles of lactate. What we did then is we extracted the DNA.
By the way, are those physiologically accurate doses? Do we believe the microenvironment of cancer is that high?
It's about 10, yes.
It's 10 times the normal level.
There's been studies showing up to 40. But normally, 10 is a typical...
And what pH?
The pH is usually between 6 and 7.
I was about to say that has to be below 7.
Yes, for sure. In fact, the more aggressive, in general, the more aggressive the cancer is, the more glycolytic is, and the more lactate is found, and the more acidic the microenvironment is. So we published an idea to propose lactate being the explanation for the Warburg effect. Because what we looked into the medical research is that the genetics stops in how the first cancer cell happens, and maybe in proliferation in cell cycle genes. But there's a lot more to that in cancer. You need angiogenesis, you need metastasis, you need immune escape, and you need also the self-sufficient metabolism the cancer cells have. It's immortal. So that's where what we saw is that lactate is necessary for each of these major steps in carcinogenesis. But what we wanted to see is could lactate also be a signaling molecule? And that's where what we observe is looking at transcriptional activity, looking at the RNA expressions of the key oncogenes, transcription factors, and cell cycle genes and proliferation genes in cancer. Lactate overexpressed them between 2 and 8-fold compared to control.
And what was the difference between the 10 millimolar and 20 millimolar lactate? Did you see a difference in transcription?
In 10 and 20, we didn't see much of a difference, but we saw more than in 0. What struck us, and that's hopefully we can show, is that if you cultivate the cancer cells in glucose alone, I give this presentation at the Anderson, but so we looked at the cancer cells and we looked at the major oncogenes, transcription factor, and cell cycle genes. We had no glucose incubation, no glutamine either, just glucose. And then we added lactate, 10 millimolars and 20 millimolars. So we did RNA extraction, and we looked at in the cancer cells where they're without any media, that is no glucose, no glutamine, we didn't see RNA expression.
Did the cells live?
Of those oncogenes. We killed them. We didn't.
In other words, there's a very finite period of time in which you're looking to just see.
Yes, we look in 6 hours and 48 hours. So neither in 48 hours was RNA expressions.
Okay, what about glucose by itself?
So when you add glucose by itself, we looked into the media, and it was a very high lactate levels.
Looks like on your graph, it's almost 30 millimole.
Yes, it was almost 30 millimole in the 6 hours. And because lactate is also used by the cells for energy purposes, over time, we expected also to see, but we still see about 25 millimolar. This is the Warburg effect. This is what Warburg observed. Incubating in cells and said, wow, they use a lot of glucose, but why in the world is this lactate?
It's amazing it's that high.
So what we saw then is this lactate alone
was enough to trigger
the expression of all the major oncogenes, transcription factors, and even depress the cell arrest genes.
So is there an experiment that could be done where you constantly change the media? You have a flux of media that allows them to have a finite amount of glucose, but you constantly strip away the lactate to see what the true baseline level of expression is, absent the lactate as the signal.
Yeah, so this is now where we're going to be replicating this experiment with multiple cancer cell lines, from liver to pancreas to lung to kidney to thyroid, more glycolytic, less glycolytic, and then do all these kinds of experiments and include also metabolomics.
This is a complicated media device because you basically have to expose them to a bath of constantly moving media that contains glucose, but no lactate. You see what I'm saying? So you have a negative flux of lactate across the cell, because what you really want to do is see how does this work with glucose, but no accumulated lactate? Because that would answer the question, is lactate specifically signaling? Because you could still argue here glucose is playing a role.
Yes, but we believe this is through the lactate.
Right, through the lactate, but now how do we figure that out?
That's when we did in the second experiment, the glucose media is the same. We just added more lactate. And we see a much amplified response.
How much is it amplified with?
So it was, for example, this is the no lactate versus the lactate. We can see
that looks like about 2x?
Yeah, sometimes even 2x. So that's where we saw that the media is the same. Yeah. But the more when we added the lactate, it really overexpressed the transcriptional activity.
So if I was going to play devil's advocate, you could say that we know that the lactate could be serving a metabolic fuel. So maybe it's conserving more glucose for more glucose to be signaling transcription.
Well, we know that lactate is being used by the mitochondria of cancer cells. Everywhere is mitochondria. there's lactate. What we believe is that it's a signaling molecule to really overexpress the transcriptional activity of oncogenes, transcription factors, and cell cycle genes in a non-hierarchical way. Because the traditional view of cancer is that you have the oncogenes, they tap on the transcription factors, and they start an array of different downstream signaling that eventually transforms a normal cell into a cancer cell.
This is so interesting because it, again, at the meta level, flies in the face of all of the observational data of how much metformin lowers cancer, unless it comes back to your explanation. Because if you just look at these data, all things equal, and by the way, that would be another interesting experiment, add metformin to the dish as well, in theory, it should amplify lactate by poisoning the mitochondria further and drive even greater upregulation of these signals, unless, to your point earlier, it becomes so toxic to the mitochondria that the cell undergoes apoptosis.
Yeah, that's what's in my opinion, and this is another thing that we want to do. But it's possibly that, yeah, because you're totally right. It can amplify the lactate, as we know, so it can amplify that oncogenic or oncogenetic signaling for carcinogenesis.
Or maybe it just doesn't matter because these amounts of lactate are so high that we're not seeing that from metformin. Maybe metformin isn't that inhibitory
so much
to the mitochondria, and that becomes a red herring in the equation, and the benefits of metformin exist totally elsewhere.
We don't know, but it will be very interesting to see all this because it can have some application, and there's some research groups studying already why.
I feel like I need to quit my job and come and be a postdoc in your lab
No.
because there's just so much. The more we talk about this stuff, and I know it's going to get way worse tonight when we have dinner with Rick because it's going to be like 50 other ideas that I just want to.
With the fructose as well. Yeah, that's a lot of things going on there.
Let's talk for a few minutes about drugs and cycling. Many people argue that the era from about 91 to 2010, there's a 20-year period of time, the 90s and first decade of the 2000s, where the use of drugs was at its highest. I don't think anybody can be a student of this sport and ever say there's been an era when the top athletes weren't using some drug. I mean, even Eddie Merckx, the greatest cyclist of them all, on many occasions was found to be using an amphetamine or something like that. How much of an impact do you think the performance-enhancing drugs of that era, the 90s and 2000s, where, again, it's all out in the open now. Everybody understands how much blood doping and how much EPO was being used. Yet you, a moment ago, gave a number of 6 to 6.5 watts per kilo as an FTP. I recall reading at the time athletes hitting 7 watts per kilo. Do you think that is about the distinction of with and without EPO? Do you think that's about the magnitude of the improvement?
I could not know that number. Yeah, I mean, definitely we know the times. And back in the days, we didn't have those power meters, or cyclists, they didn't use them. So it's difficult to calculate, but it is possible to calculate with the times. I haven't done the numbers, but what we know now is that the times going Tourmalet, Alpe d'Huez or so are the same times that people were doing in the 80s or early 90s. It's hard to see any of today's cyclists being in the top 20 best times now as they did before. So that's something that shows that, yeah, cycling, thank goodness, is a very clean sport right now. The other thing is the fact that every cyclist now who wants to do well, they go to altitude. And that's one thing that before didn't happen. And now it's great to see people going to altitude because it's a physiological way to increase oxygen carrying capacity.
Is the data on altitude still the following? So when I last looked into this, which is maybe a decade ago, the answer seemed to be the performance-enhancing way to use altitude is to live high, train low, meaning your baseline exposure should be at a low oxygen environment. Your low-intensity training should be at a low-altitude environment, but your high-intensity training should be at sea level. Is that still believed to be the case?
That's an ideal scenario, in my opinion, yes. And I mean, here in Colorado, because we're Colorado here, by force, we must know about altitude because we get a lot of athletes every year and we get to study them. And yeah, one thing that happens at altitude, your glycolytic capacity, it starts deteriorating for high exercise intensity. It's like if you had a cap here at altitude. And this is something that everybody tells you when they're here, like, I cannot give my 100% here. I cannot open up the same gas as I used to have. And if you don't do that for 3.5 weeks or 3 weeks at your altitude, your glycolytic capacity is going to deteriorate, which for a marathon runner, couldn't care less. But for a cyclist, for example, it's important. So that's where the ideal scenario is to really try to find that balance, but it's not easy to do at high altitude levels.
Has anyone ever proposed using little portable oxygen producing devices for peak efforts for those who live at altitude to maintain top end?
Exactly. And that's done.
It is done.
Yeah, it is done. Yeah. And that's something that we're building at the university in one of our campuses in Colorado Springs, a sports medicine and performance center, where we're going to have one room that is going to simulate sea level conditions. So it's going to be hyperoxic. So it's going to simulate living at sea level, because that is going to allow athletes to do very high intensity efforts without killing themselves. Because this is the problem that happens here at altitude. These athletes who want to do very high intensity exercise, which they really need also too, they get overtrained. We see a lot of people living altitude in very fat form, and they're overtrained, or they live with a high oxygen carrying capacity but poor glycolytic capacity. So that's where by doing this space, it's going to allow athletes to come and they use these facilities while living at altitude. Because the problem that we have here, you have 2 days driving to the ocean, so you cannot train low and live high. But at least train low, high intensity, you can simulate that while still living in high environment.
So interesting. Miguel Indurain, have you ever met him?
Yes.
An unbelievable specimen?
Yeah. And an incredible person too.
I've heard he's got to be what now? He's got to be 60.
Yeah, 50s, in the high 50s or so.
50s, yeah. Still incredibly fit. I remember reading a paper about him maybe 20 years after he retired. He retired in 95, 96, right? I mean, still unbelievable numbers. I mean, was he just a physical phenom to begin with?
Yeah, he was incredible. When I was doing my internship, I was doing it with an endurance physiologist who was a very, very good physiologist, and I learned a lot. And I remember once I was kind of helping there. And one thing that struck me also was his numbers were unbelievable, and also the amount of sweat that he had. I have never seen anybody sweating so much in my entire life.
He was a big guy, right? He was
big
80
guy.
kilos,
Yes.
6'1", 6'2".
It was incredible, because usually when you do a physiological test, you might have one towel or a couple of towels. Even with fans, people sweat a little bit. And back in the days, I was just wiping the floors. That's kind of, you do internships. In these cases, with Indurain, you needed a mop.
And have you seen athletes since?
Never.
So do you think that's just true and unrelated, or do you think that also spoke to his physiology? Like he had an unusual cooling system?
He was out of this world. I have never seen anybody like him. And if you observed in the Tour de France, everybody was with their shirts wide open, right? And Indurain was always all zipped up, always, and with a hat on. So he had an amazing capacity to dissipate heat, which is a double-edged sword. So obviously he drank a lot, but I'm very sure, and back in the days we didn't have the technology that we have nowadays to measure that. What we do now, sodium concentration in sweat, we do sweat tests. We have sweat patches, and we can measure the sodium concentration in the sweat patches and then tell someone, whoa, you're a heavy sweater in the first place, and you also sweat a lot of sodium. But when someone, and it's something that's very typical, we see in young people, or people who are not very well adapted to sweating, you see the white marks in their shorts or in their helmets. That's salt, literally, sodium. But the more mature physiologically an athlete gets, the more they sweat.
That is so, I have never realized that is a metric, yet another little trick of the trade to look at sort of metabolic flexibility. Is the ability to retain the sodium as just the water leaves.
Yes, exactly.
Makes total sense.
Yeah, it's an evolution. And you sweat more. Back in the days, I was just wiping the floors. But
now you'd bet that there's low sodium in there. You'd pull a William Osler, and he figured out that diabetes urine tasted like sugar. You'd figure out that Indurain sweat tastes like water.
I would have tested it for sure. I guarantee you. I would have said, man, there's no salt here.
Indurain's interesting because he's right on that precipice where there is no question that the person who won the Tour right after him was using Herculean doses of EPO. So Bjorn Ries won in 96, nicknamed Mr. 60, right? I mean, hematocrit somewhere between 60 and 66. So you go from Bjorn Ries to Ulrich to Pantani to Armstrong. That's the era. And then before Indurain is Greg LeMond, who, again, I don't really know anything about what he was or wasn't taking. But Indurain has largely been left out of the discussion on blood doping. And I've read articles that have just talked about how he's generally been left alone. No one has come back to him so I'm not going to ask you to speculate on that because I know that from a personal standpoint, I don't want to put you on that spot. But do you get the sense that he's just been left out of this discussion because of his place in cycling, and it's almost like people don't want to go back and revisit that? I mean, why do you think that is?
I don't know. I have no idea, to be honest. I could never give you an answer. I know that he was a freak of nature because of his size. In the same manner that before Indurain was Greg LeMond, was also
a freak of nature.
A freak of nature as well, and left out of all this. I have no idea, but I've seen his physiological parameters, and I've seen a lot of athletes. You don't see those physiological parameters. And also, what I always say about Indurain is his head. And I work with a lot of athletes. And in cycling, for example, I have never met any athlete. Well, there's one athlete. I will tell you in a few years.
Meaning there's someone you work with now who maybe in a few years you'll tell us.
Yeah, I don't want to be...
No, you don't want to jinx him.
Him too, yes, exactly. But only one athlete with his head. He was calm, he was relaxed, he was super intelligent, he could read the game ahead of things. He would never get nervous about anything, and he would never doubt about anything, which is rare in athletes. I've seen athletes getting to the top of the game and falling apart and start crying. There's the fear to lose, but also the fear to win. Because when you win, your life changes, for the good or for the bad. And many athletes, they were always nervous, trying to find an answer or trying to find a new diet or a new training or something, and that's where we're very fragile. Athletes at the high level are very, very, very fragile. If you're considered like an expert or you're a coach or you're someone with a little bit of a name in cycling, for example, and if you go to a race and you see a cyclist, wow, you look fat, I think you gain weight, that cyclist is done.
Yeah, exactly.
Done.
I mean, cyclists are like models in that regard, right? Every ounce matters.
Exactly, and that's how they are. But Indurain started the tour with 2.5 kilos over, for example. Why? The first weekend the tour is flat, no gravity. His head is relaxed and calm. They're like, okay, I can do this, no problem. And in that week, he loses a kilo and a half. And then he entered the second week with the mountains with half a kilo over. Okay, no problem, no big deal. He loses it, and then boom, the last week is in perfect weight. It takes a lot of thinking and
confidence.
And confidence, right? And saying, hey, I got it. And that's what I think that his head was also unbelievable. LeMond's head was also incredible. I remember as a kid reading LeMond's book. It really literally changed my way of looking at cycling. I was 15 when he won the Tour de France. And that's where I started to see his complete book of cycling. I don't know if you've ever read it.
No.
It's the best cycling book I've ever read in my life. And it's about how he trained and how he ate and how the way he approached cycling. And back in the days, I'm talking 86, he was super scientific.
Yeah, he was so far ahead of
everybody else.
Yeah, we could go on for hours. Yeah. But I think on this note, we'll bring it to a close only because we're already late for dinner. And I don't want to keep Rick waiting. But I want to thank you so much for this. This has been incredibly informative. There's a million follow-up things to do, including we're going to do this biopsy study. So I'm willing to come back to Colorado to do this again because I really am curious about this metformin question around zone 2. And I think that this is going to be one of those episodes where hopefully people are able to see the show notes because so much of what we've talked about, I think, benefits from this type of being able to visually see what this stuff we've talked about. And lastly, I do think there's going to be no shortage of medical students and undergraduate students who are looking for summer projects to come and help you get a lot of these really interesting posters published. So thank you.
Well, thank you so much, Peter.
For the impact you've had on me personally with respect to how I think about this problem, and then hopefully by extension how others have as well.
Well, thank you so much. It's truly an honor to have you here and to speak with you and be invited to your podcast. Thank you very much.
You can find all of this information and more at peterattiamd.com/podcast. There you'll find the show notes, readings, and links related to this episode. You can also find my blog at peterattiamd.com. Maybe the simplest thing to do is to sign up for my subjectively non-lame once-a-week email where I'll update you on what I've been up to, the most interesting papers I've read, and all things related to longevity, science, performance, sleep, etc. On social, you can find me on Twitter, Instagram, and Facebook, all with the ID PeterAttiaMD, but usually Twitter is the best way to reach me to share your questions and comments. Now for the obligatory disclaimer. This podcast is for general informational purposes only and does not constitute the practice of medicine, nursing, or other professional healthcare services, including the giving of medical advice. And note, no doctor-patient relationship is formed. The use of this information and the materials linked to the podcast is at the user's own risk. The content of this podcast is not intended to be a substitute for professional medical advice, diagnoses, or treatment. Users should not disregard or delay in obtaining medical advice for any medical condition they have and should seek the assistance of their healthcare professionals for any such conditions. Lastly, and perhaps most importantly, I take conflicts of interest very seriously. For all of my disclosures, the companies I invest in and or advise, please visit peterattiamd.com/about.